There are my disclosures. So I needn't tell this audience about the need for safer and better therapies for lupus, but developing drugs in lupus has been a difficult challenge. And the reason for that are many, and I've just listed a few here. So one is the heterogeneity of manifestations will complicate trial design. And though we don't have many different background medicines to choose from, we certainly have confounding by these background medicines, especially the steroids. And trial endpoints have been difficult as well, though that nut we have probably cracked. So despite all these challenges, there's unprecedented SLE trials activity at this point in time. So what should be the target? This is a busy slide, but I'll make it a little simpler. And this is my simplistic explanation of the pathogenesis of lupus. So in a genetically susceptible host, there's an environmental trigger. In this case, the sun, which induces apoptosis of skin cells, the release of DNA, which activates the plasmacytoid dendritic cell through the TLR receptor. Then there's the elaboration of a variety of cytokines, chief of which is interferon alpha. We'll cross over to the adaptive immune system and involve the myeloid dendritic cell, the T cell, as well as the B cell. And my focus in the rest of the talk will be on B cell targets. But all of these components of the immune system have or will be targeted pretty much for lupus drug development. B cells are dependent upon APRIL and BLyS for survival, growth, and differentiation. And B cells, as you know, can differentiate into plasma cells with the elaboration of antibodies. And in the case of lupus, autoantibodies to DNA. The complex with DNA is capable of activating complement, attracting neutrophils, and activating macrophages. And this is a self-perpetuating cycle where the plasmacytoid dendritic cell will be activated through the Fc receptor. So again, focusing on B cells, there are different ways to target B cells. And I've divided it into cellular targets as well as extracellular targets. In this slide, we have a lot of potential targets. But let's erase the extracellular targets and then concentrate on CD20 and CD22. The pre-study expectations with Rituximab were amazingly high. We just thought, do a couple of studies. We already know the drug works in lupus, and we'll have another drug in our toolbox. But it wasn't that easy. And our pre-study expectations were based upon off-label experience as well as open-label study results. But the randomized controlled trials, Explorer and Lunar, were both failures. And then along came Ocrelizumab, a humanized anti-CD20, and the Belong trial, which is a trial performed in lupus nephritis patients, was terminated prematurely because of opportunistic infections. But is B-cell depletion therapy dead? I don't think so. Clinicians still believe. And what is this based on? In part based upon Lunar data. If one looks at week 78 proteinuria reduction, one actually sees a statistically significant result. There were far fewer cyclophosphamide rescues in the treatment group. In fact, it was 8 versus 0. The effect size was no different than in BLyS 76. So maybe all Lunar needed was more patients. So Rituximab definitely remains in our armamentarium for refractory nephritis, for autoimmune cytopenias, for CNS disease. And the interest remains high, certainly in Europe, where studies are ongoing or soon to start. But will companies pursue an indication in lupus? I don't know the answer to that. Let's turn to anti-CD22, Epratuzumab. It has an immunomodulating effect and only results in partial B-cell depletion. There was an early study of 14 patients which served as the foundation for a large phase 3 program. Unfortunately, the phase 3 program was terminated prematurely for a lot of different reasons. But that particular study served as the foundation for a phase 2b study known as Emblem. And Emblem was a success because it reached an endpoint as early as week 12. It also introduced a rigorous composite endpoint known as BICLA. Partial B-cell depletion was seen, and a phase 3 is well underway at this point in time. Now let's turn to the right-hand side of the slide and focus on extracellular targets. Bliss and APRIL are key growth, survival, and differentiation factors for B cells. Bliss binds 3 receptors, Bliss receptor, TACI, and BCMA. APRIL binds just the bottom 2. There are different ways one can inhibit Bliss and APRIL. So one way is with a recombinant receptor, TACI, or atacicept. Another is with a peptibody, blisbimamide. And there are 2 monoclonal antibodies out there, to belimumab as well as belimumab. And we're going to concentrate on belimumab. So why even target Bliss? Well, as I mentioned, it's crucial to B-cell maturation, differentiation, and survival. Years back, in murine models, transgenic mice for Bliss developed a lupus-like illness. And it was shown that TACI-Ig could reduce disease activity in these murine models. And certainly in human lupus, there were observational studies demonstrating elevated levels, as well as the fact that high levels were predictive of flare. The phase 1 study was done, which focused on pharmacokinetics, pharmacodynamics, and safety, and that served as the foundation for phase 2, which focused on efficacy. Though the phase 2 study was a failure, not reaching its co-primary endpoints, there were a lot of clinical signals, there were biological signals, there were no safety signals, and what came out of that was also the responder index, known as the Sle Responder Index, or SRI. In order to be considered a responder, one had to have a 4-point improvement in the Selena-Slide score, and no bilag worsening, and no worsening in the physician global assessment. So this served as the endpoint for the phase 3 program, and you've seen these results so far in the meeting. The primary endpoint, the SRI at week 52, was met in both phase 3 studies. This led to FDA approval in the United States about 2 years ago, and now approvals around the world. But since that approval, there have been a lot of questions. So how does one apply the data to everyday practice? And here's a whole list of questions that we'll address in the closing of the talk. My RA colleagues asked me, why was the effect size smaller than in the rheumatoid arthritis studies? So the effect size in Bliss 76 and Bliss 52 were 10 and 14%. If one raises the threshold of a response, certainly in Bliss 76, you can see a slight increase in the effect size, up to about 12%. If one looks at patients with high disease activity, those with low complement and high DNA antibodies, the effect size is as high as 20%. Why is the effect size smaller in lupus studies than in rheumatoid arthritis studies? Well, the SRI represents a very high threshold of response. It's a dichotomous instrument nested in a dichotomous instrument, meaning it's an all or nothing response. The SRI does not capture partial response, nor does one of its components, the SLEDAI, capture partial response. So, in summary, it's a very difficult threshold to achieve. I also think that the greater the number of partially or fully effective background therapies, the smaller the effect size. And in the Bliss trials, approximately 2/3 of patients were on 2 or more background therapies. And it may be that specific subsets respond better than others. Commonly asked question is which manifestations respond to belimumab therapy? And this is from an article by Sue Manzi showing which SLEDAI domains responded: mucocutaneous, immunological, vascular, and CNS. But be careful, these are subset analyses, and the study was not powered for these subsets. What are the effects in lupus nephritis? The study excluded patients with very severe lupus nephritis and those with 6 grams or more of proteinuria. But still there were some signals which came from the Bliss trials. It turns out that about 16% had SLEDAI renal domains, and if you go by BILAG A or B, 11% had renal activity. 20% had proteinuria greater than 0.5 grams, and 6% greater than 2 grams. And you can see on the bottom of the slide some of the clinical signals. Fewer renal flares in the treatment group, greater number of remissions in the treatment group, and greater declines in proteinuria in the treatment group. And this prompted a lupus nephritis trial, which is underway at this point in time. Do African Americans respond? This was a very contentious issue at the FDA Advisory Committee, and I actually will say no more because this will be addressed in our next talk. How early do subjects respond? Well, it definitely depends on which metric one is looking at. If one looks at the SRI in the combined BLISS 52 and 76 trials, one sees responses at 16 weeks. But looking at other metrics, one sees responses that occur earlier in time. Fatigue at 8 weeks, and serologic changes as early as 4 or 8 weeks. What happened to corticosteroid dosing during the studies? And I should remind you there was not a mandatory taper. But in those who were on 7.5 milligrams of prednisone or greater at the outset of the study, you can see results here, both in the pooled trial population as well as in patients with high disease activity at baseline. There was numerical superiority for those who received belimumab treatment. And you could look at the reverse. What percentage of patients had to increase their steroids? These are in the high disease activity patients, and you see 41% versus 29% had to increase their steroids, placebo versus treatment. This slide shows the effects on serology. That is normalization. Greater numbers in the treatment arm normalize their DNA, normalize their C3, and normalize their C4. And this slide shows the effects of fatigue, with changes occurring as early as 8 weeks. Are the effects durable? Well, to address that, I've gone back to the phase 2 study. At the end of the phase 2, and this was a post hoc analysis where all treatment groups were combined and the SRI applied. And you see on the left-hand side of the slide, 46% achieved a response versus 29% in the placebo group. And that response was carried out because the placebo patients and the patients treated with the lower doses of belimumab, 1 and 4 milligrams in the phase 2 study, were all converted to high dose, 10 milligrams per kilogram. And you see durability on the right-hand side of the slide. We're up to about year 8 or 9 in the extension study, but these data show up through year 7, with response rates in the 60% range. Another way of looking at it is flares. Again, this is the phase 2 extension data. So all flares, you see a decline over time. On the left-hand side, the double-blind portion with about 85% of patients having some type of flare, and then a reduction over the 7 years. And on the bottom, severe flares, going from close to 20% during the study down to the low single digits in the extension study. What does an SRI response really mean? It's a little hard to get your arms around what it translates into. So Vibeke Strand and I put together this information. And we basically asked the question, forget about treatment assignment. Just look at patients who responded and compare them to patients who were non-responders. And we looked at a lot of different manifestations, which are listed here, and we'll just concentrate on severe flare rate. So 6% who were responders had severe flares. 4 to 5 times that in the non-responder group had severe flares. Fewer increased their prednisone who were responders. There were more favorable changes in serologies. And then shown are the SF36 and the fatigue numbers, both favoring SRI response. So SRI response translates into clinical benefit in many different manifestations. What does the future hold? There are a lot of drugs in development. In fact, I say there are more studies than there are patients. And that speaks to some of the issues that I addressed earlier today. We need to become more innovative with our design. But the development and approval of belimumab will hopefully pave the way for yet more therapies for our patients with lupus who sorely need more effective and safer therapies. Thank you.